Hi, everyone. Welcome back to the AMPD webinar. We're going to start part 3 now, which discusses using the Terra Workbench. This part will be addressed by Allie Hajian and Barry Landon. Allie Hajian is a science writer at the Broad Institute's Data Sciences platform. She develops documentation for Terra and is helping researchers transition to a cloud-native platform that can accelerate their discoveries. Barry Landon is the solutions architect at Technome and the data coordinator for AMPD. He is helping to bring systems and data together in a way that is easy to use and easy to share. So we're going to start with Allie.
Thank you so much for the introduction, Deb. Welcome everyone to part 3 of the webinar. Can we have the next slide? So here's an outline of what I'm going to cover in the next section. Barry and I are going to cover, in part 3. I'm going to give a brief overview to the Terra platform, which is the structure that is in between the cloud and the data in the cloud and the researchers. So I'll give an overview. I'll show you some of the features, including the data library and some getting started workspaces. And then after I give that introduction, I'm going to do a hands-on exercise in a quick start workspace that you can all have access to. If you've registered for Terra and you're able to log in and you've taken advantage of your free credits, you'll be able to follow along with me. If you haven't registered yet, it's okay, because the quick start workspace is pretty self-explanatory. So you can watch along while I do it and then go back and do it yourself. So hopefully I'll give you everything that you need to know. When we're doing the hands-on part, there's 3 basic steps. The first, we're going to go to the data library and we're going to use the Data Explorer to access some data. We're not going to use AMPD specific data. We're going to use some public access 1,000 genomes data, but it's very similar. So I'll demonstrate that. And then we're going to take that data and put it into the workspace in a data table, an integrated data table, and we're going to analyze it in an interactive Jupyter notebook. And I'll show you how you can work with the data. And then the last hands-on part I'm going to do is to run a workflow or a pipeline on Terra. So I'll explain the vocabulary a little bit more. And then when we've done that part, I'm going to hand off the rest of the session to Barry, who's going to show you some additional resources in Terra about workspaces, billing and cost management a little bit, and then some of the quick start workspaces that are specific to AMPD. So can I have the next slide? Okay. Sorry, mine isn't going forward. So in the previous session, you heard about some of the advantages of doing research in the cloud and the way we're organizing and consolidating the AMPD data to make it available to researchers. And Dave also talked about the Google Cloud components and how those work together for helping analyze and store and access the data, and some of the web resources. And I'm now going to talk about Terra, which is a cloud native platform that allows you to access data, run in the cloud, run analysis tools, and collaborate on biomedical research. And it's an interface at its heart. It works on top of the Google Cloud platform, and it's designed to bridge the gap between resources that are in the cloud that Dave talked about and the researchers, to make working in the cloud a little more intuitive and hopefully a little easier and quicker. So the first things I'm going to do is talk about at a very high level about some of the features of Terra that can help you in your research project. And then we're going to dive in and do some hands-on exercises. And if you have registered for account on Terra, you'll be able to follow along with me. You can go ahead, you can log in if you want now, or you can wait a few minutes till I'm done with the slides. And can I have the next slide? So before I start, I want to acknowledge that building a platform like Terra takes a lot of collaboration. The Researcher Workbench that you're going to see, is a generic life sciences platform. We host many data sets and we work with many projects, and it's being built out currently by the Broad Institute of MIT and Harvard, and Verily, which is the life sciences part of Google. So, history of Terra. So, Terra started around 2014, so not actually that long ago, when the NCI selected 3 different pilot groups to bring over about 2.5 petabytes of data to the cloud. So, this was a very cancer-specific project, and the Broad, the Institute for Systems Biology, and Google worked together to bring that data and the tools to Google Cloud in a platform that we called FireCloud. But as FireCloud continued to evolve, it was pretty clear that what we were building wasn't actually specific to cancer research, but could be used more broadly for anything that involved genomics data, and actually anything that involved big data in Google buckets and BigQuery. So we started developing a second platform, which Terra, which was intended to be more broadly applicable. It's built on the same back end, so it's very robust, but it has a lot of additional things that features that Terra did not have, that extends it beyond that first use case for data. And what that means is data sets like AMPD are accessible and analyzable on the Terra platform. So what does the platform actually look like? Can I have the next slide? Okay. This is a screenshot not of the landing page, but of a workspace in Terra. And a workspace is a fundamental unit that is where it's like a computational sandbox that keeps everything you need for a particular research project in one place. It's secure, it's easy to share, and it contains the data, documentation, it has the data, it organizes the data that you want to include. It has analysis tools, notebooks, and workflows, which are pipelines that do work in the cloud. And it also keeps track of job history, so that you know exactly what you did and when and what the results were. So if you need to reproduce or you want to go back. So that's a very big overview of the workspace. Can I have the next slide? What other things are on the Terra platform that you can pull into your workspace? The first thing is data. Terra hosts a number of data sets. There are both public access and restricted access contained in the data library, and all of you are probably familiar with the one on the upper right. We will go into the data library when we do the hands-on portion, but it has several data sets, and this is what it looks like. Can I have the next slide? If you were to browse one of the data sets in the data library, you'd get directed to built-in functionality for exploring, and this allows you as a researcher to create a customized subset from the data in the data library. This is what Dave was doing when he was defining the inclusion criteria. It's the same idea, except you're using a graphical interface for doing it rather than doing a SQL query. And we will do this in the hands-on portion, so you'll get you'll get an idea. This particular screenshot is for AMP-PD Tier 1 data, and if you look, it should look like it's similar to what Dave talked about and what you went through on the Google Cloud Console platform. Can I have the next slide? So another part of the Terra platform is integrated analysis tools. There are several different analysis tools available within the Workbench platform. These are three of the forefront examples. HAIL is an open source scalable framework for exploring and analyzing genomics data, can generate variant and sample annotations, and perform variant gene burden EQTL association analyses. That was developed at the Broad. The second is a scientific workflow engine designed for simplicity and scalability called Cromwell, and its mascot is the James the Pig. It has a background that you can read about on Terra. And this is what's used to access the Google Compute engine in the cloud. It's what goes between the Workbench platform and the Compute engine. And I'll talk a little bit more about that when we actually use it. And the third integrated analysis tool is Jupyter Notebooks, which are interactive development environment for working with, Terra has an interactive development environment for working with notebooks, code, and data in Jupyter Notebooks, and we will go through that as well. There are several notebooks on the Terra platform that are available, template notebooks that you can use, and I'll talk about those, and specifically the AMP-PD ones that exist. Can I have the next slide? Okay, I'm going to go into a little more detail on the two different kinds of analysis in Terra. The first is interactive analysis in Jupyter Notebook. According to the jupyter.org website, Jupyter is an open source web application that allows users to create and share documents containing live code, equations, visuals, and narrative text. And we're going to do a hands-on exercise that'll show this rather than telling you. But because notebooks contain all of those components, it's a really great way of sharing with people. You can share a notebook and your collaborators can see exactly the code that you ran. They can run it as well, and they can understand with the documentation what you were thinking and why you made it. And we use Jupyter Notebooks. In contrast, there are other notebook environments, but Jupyter is open and interoperable, so it works well with the Terra platform, which is also open source. The second thing that is really nice about notebooks, in addition to being able to share your analysis and your results so easily, is you can visualize and iterate in real time. As you'll see, BigQuery, because it's optimized for querying, even with really big data sets like AMP-PD transcriptomics, you can run analysis in a notebook instantaneously, well, almost instantaneously. You can see the results. If you want to tweak it, you can go back and redo some of the code and rerun the results. So it's very powerful in that it's interactive and allows you to do your analysis in a really iterative way. So we are going to import and analyze data from the data library in a Jupyter Notebook in the hands-on portion, coming up in a few minutes. Can I have the next slide? So another tool, analysis tool in the Terra Workbench is automated analysis with workflows. So workflows are pipelines. They are batch submissions by Terra's Cromwell execution engine to the Google Compute in the cloud. They're often used for preparing and processing genomics data. For example, aligning reads and calling variants, and you will notice that some of the tools that are in the Terra code and workflows library might be familiar if you've done some of that work. We have joint discovery tools. We have a somatic panel of normals workflow, variant discovery tools, and if you don't find what you want on Terra, there's also a Broad Methods repository and doc store where additional tools are available that are easy to import into your workspace. And you can see that in the panel on the right. So at the very end of the hands-on portion, we're going to run a very quick workflow on sample data. So I'll demonstrate how quickly it can go in the Terra platform. Can I have the next slide? Okay. There's a lot of resources, and you can certainly have the opportunity to build your own workspace from scratch in Terra. Terra also has a number of curated templates that we call featured workspaces. They span a variety of use cases, and they're really helpful for getting you started on the platform. They're standardized, so they all look the same and have about the same components, and they're meant to be straightforward and easy to use, so you can reproduce the results, and you can learn how the established ways of doing things. There are two getting started with AMP-PD workspaces, one for Tier 1 and one for Tier 2 access, and there's several others in development, which Barry will talk about after this portion. And they have built in already some analysis tools that you'll be able to adjust to your own research. Can I have the next slide? That's my general overview, and this is an outline of the next portion, which is hands-on accessing and analyzing data in the cloud. And we're going to do 6 things. I'm going to have you log into Terra if you aren't already. And I'll give a quick overview of where things are. And then you're going to clone your own quick start workspace. And we'll see some of the pieces that are there. We're going to take a side route to explore some data in the Data Explorer and push it to the workspace that you just created. And then we're going to take that data, analyze it in a notebook, and I can show you how that works. And then the last part, as I mentioned, is we're to run a batch analysis workflow. And I'm hoping that it will finish in a few minutes, and you'll be able to see it finish. So, apart from logging into the platform and looking at documentation, all of the things in the list above require us to pay Google. It's a small amount for the Quick Start workspace, but it's something. And what enables you to try the platform without a big investment is something we call free credits. Oh, can I have a next slide? So free credits are, they give newcomers to the platform an opportunity to try it out at no cost. And when you initially log in, when you create your account and log in, you will get a pop-up that will ask you to sign up for free credits. You need to accept the terms and conditions, and then you'll see the free credit billing account in your billing section, which I'll show you where that is, and you will be the owner. The free credit program begins when you accept the terms and conditions, and they expire after 60 days or after you've used $300 worth of credit. So if you have signed up for Terra, and if you're able to log in, that would be great. You can go ahead and go to your Terra account and follow along with me. And if you haven't logged in, or if you haven't accessed your free credits, you can certainly do so now. I'm going to let us have a pause. If anyone has any questions about the free credits or about logging into Terra, you should ask on the chat while I switch to the hands-on portion, and I will open my hands-on window. So for the hands-on portion, if you can go to the landing page for Terra, app.terra.bio, you will see this as your, I'm going to try to get rid of there. This is the landing page. Welcome to Terra. Right here are documentation, tutorials, and discussion. And the main navigation menu is at the top left here. So if you click on the three horizontal lines, you'll get, you can access your profile, your billing. If you click on billing, your billing project should show up here. Yours will look very similar to this. It'll say FC credits, and then it'll have a metal and a color and a number. There's groups information, which has to do with some of the structure within Google, and notebook runtimes, which I will talk about in a second. You can also access your workspaces and the Google library, the Terra library, of which there are three. There's a data library, a showcase workspace library, and workflows. And what I'm going to lead you through first is going to the showcase library, and then we'll take a break and go to the data. The last thing on the last item on the main navigation menu, which is on every page, is support functions. So here's documentation, request a feature. Community forum is where all the members of the community go to post questions and answers, and then a contact us. So those are all the things that are in the main navigation. So if you go now to the showcase workspaces, on the left are GATK4 workspaces. These are the best practices, workspaces for the GATK tools. Each of the featured workspaces has everything you need to get started. So it would have documentation to explain what's in the workspace. It will have sample data so that you can run on sample data if you want. It has the workflows that you need. I'm going to click into one just to demonstrate. So here's a little background. What are the workflows? What do they do? What kind of input do they require? What is the output? What's the reference data? There are step-by-step instructions on how to use the workspace to do that kind of analysis. There's a more detailed description of the data. So this one has a subset of the chromosome, which you can use to test, but to make sure that the workflow runs correctly. And it gives time and cost estimates, so you can have a handle of, before you run it, of how much it's going to cost you in your Google compute time to run. And then there's some contact information for us in the licensing. The data are in here. These workspaces don't have any notebooks because they're workflow specific workspaces. So this one has one workflow. It's variant calling using a Spark multi-core, and then the job history tells you if gives provenance for when you ran a workflow, what you ran the workflow on, did it finish, did it fail, when, and that all that sort of information. So if we go back to the showcase library, I would love for you to click on the Terra Quick Start Workspace. So this is a workspace that if you read it and run through it all, and we're going to run through a good portion of it, it will give you all the experience in the basic functionality of Terra, and hopefully give you confidence to run your own workflows or to run your own notebooks. And this is what we're going to go over in the next 20 minutes or so. So in order to do anything in a workspace, you notice at the top this says read only. You need to make a copy that is your own, and you'll be the owner. It is stored in your dedicated Google bucket. And the way you clone a workspace is by going over to the three dots over at the right and clicking on clone. This is also how you share a workspace, and in the future how you would publish a workspace. So if you click on clone, a modal comes up. You need to rename the workspace, and you should name it something that makes sense to you. I'm going to name mine with my initials and the date. For billing project, if you're new to Terra, you probably only have your free credits. If you have other billing projects that are associated with Terra, they'll also be listed here. And the last part right here, the authorization domain, is the way that Terra makes sure that you're only sharing and copying things that you have that you're authorized to do so. So this is a public access domain. There's a public access workspace. There's no restrictions on who can see it. If there were, for example, the AMP PD workspaces, you would need to have AMP PD authorization domain criteria here. So if you're following along, you should go ahead and name your workspace and select the billing project, and when you do that, the clone workspace button will become active, and you can go ahead and clone the workspace. I'm not going to do it because I made my workspace earlier today and I don't want to have too many of them. So you should go ahead and do that. And when you do, you'll be taken. I'm going to go into my workspaces, but yours will come. When you clone it, you will be redirected automatically to your cloned workspace, and it will look just like this. Okay? So, what I'd first like to direct your attention to is the dashboard has a lot of documentation. This is the dashboard is in Markdown language, so in Markdown, so you're able to have some nice formatting that lets you distinguish pieces. And this is where you would explain what your project is about. Where does the data come from? What's the rationale for what you're doing? What tools, what workflows, what notebooks are in the workspace, and you can edit by clicking on the edit button right here. Also on the dashboard, the front page, is some useful information like who owns the workspace and when was it last updated. And at the bottom of the right-hand column is a URL for your Workspace's dedicated Google bucket. So every workspace that's created on Terra gets a Google bucket for holding data. When you generate data in a after a workflow analysis, it automatically gets written to the Google bucket, and we'll go through that. But it's useful to have right at the bottom here the full path of the Google bucket, and you can, by clicking on the icon at the right, you can copy the bucket. So at the top right, there is this interesting feature that is actually related to the notebooks that I'm going to have you, if you're working along with me, I'm going to have you actually start up. So you can go to the little gear icon at the top right, and you can see that notebooks have a lot, can have a lot of different options. So when a notebook is running, Terra creates something called a runtime environment in order to do the analysis in the cloud. And the runtime environment is actually it's a combination of the virtual machine or machines or cluster that you're going to be running in the cloud, as well as libraries and other pieces that are useful for whatever analysis you're running. And there are several default environments, and we're going to run the default environment that is included in Terra. If you wanted to do a custom environment, you can specify your own custom. Will have different costs. You notice that before you even set your runtime or go out and create a runtime, Terra will give you the cost. It will tell you how much it's going to cost, and that cost is running no matter what is going on in the virtual machine that you're running on. But Terra does have a safety valve, so if you are running a notebook and you are not working and there's no analysis going on, Terra will pause after 20 minutes, and so that you're not getting charged. And this is all very similar to what Dave was talking about. He created a virtual machine running in the cloud using the console, using the terminal, excuse me. This Terra does it for you. So the default is good. You can hit replace, and then what I'd like for you to do is just hit play before we move on, because what is happening when you hit play is Terra is creating your virtual machine to do your analysis, and it takes a few minutes. So we're going to go back and do some work in the data library while that is creating and running. So if you're working alongside me, you should go ahead and hit the play button that will start your notebook, and you can just set that aside, and it'll be creating its runtime while we do other things. So the next place we're going to go to is the Data tab, and the Data tab is the place where you're going to organize all your data. Some of your data may be in the Workspace Google bucket. However, as Dave talked about, one of the advantages of using data in the cloud, like AMP-PD data, is you don't have to copy and store it all in your own workspace. You can reach out, you can reference with metadata, data that someone else is paying to store, like AMP-PD, and that's a great feature. But you still need to keep that data organized. You still need to know what data your project needs. And in Terra, the way we do that is with tables. And the workspace as it is right now does not have very many tables. It only has 2. And if you click on the tables, you see that it's two rows which are containing two data samples. We know it's a sample because it's called sample, and I'll go through that. And the first column is a sample ID. The second column is a data file. And if you hover over it, you see that this is actually in a public Google bucket. So the file is elsewhere, but all the metadata is contained in Terra. And when we run a workflow, you'll see how the data model or the data table gets updated. So as you do your analysis, it will add all the data that you can have created and keep it in the same place. So you notice that as it is now, we only have the sample data that was included in the original workspace. So let's go into Terra in the data library, and let's add to the data that's referenced in our table. So if you go to the main navigation menu again, and this time let's pick the data library, and the one over here on the right should look familiar to you. And if you've registered with the AMP-PD portal, you'll have access to either the Tier 1 or the Tier 2 data. And but for this exercise, because we want to make sure it's available for anyone, we're going to use public access data, 1,000 genomes. It's going to be very similar. So if you click on the Browse Data, you'll be taken to the Data Explorer. And this is the interface. This actually looks very similar to the Data Explorer that you would get with the AMP-PD data, except that, of course, the parameters are different in the two data sets. So this is data that's in BigQuery. So it's in the cloud in BigQuery, and these are the different parameters that you would use to include or exclude participants in your study. So as part of the exercise, I would like everyone to go ahead and select some kind of make up some inclusion criteria for your study. Say you only want to study Africans, you only want participants that have an exome cram, and we want people with a lot of coverage. So I've selected some inclusion criteria there, and if you notice up at the top, it will now tell you how many participants in the overall data set are included when you put those restrictions on. So for the cohort that I've created has 250 participants. So you should go ahead, if you could go ahead and just make a few selections, and note the number that's in the study that you've made. The one thing I will suggest, because it will make the analysis we're doing a little bit more interesting, is don't select by gender. So don't select only men or only females. And then when you're done generating your custom cohort, you can save it. And again, you want to name your cohort. It's useful to name it something that makes sense, and that when you go back after maybe not having looked at it in a while, you'll be able to say, oh yes, that's the cohort that I did in my AMP-PD webinar, or whichever you did. And then if you click Save, it'll take you to this screen. You can select a destination workspace. You're going to select the workspace that you just made earlier, and you click on Import, and you get taken, automatically redirected back to the Data tab. And if you notice now, there are a few extra tables of data in your workspace. So the cohort table right here, if you click on that, it gives you some information. So here's the cohort ID. This is the cohort that I just created. The data set name is what the library was. This is the URL to the Explorer. And then the last column is the actual query that represents the cohort that you made. So when Dave was doing this in the cloud, he actually wrote in SQL his query, but Terra does it all for you. So if you triple click on it, it will select the whole query if you wanted to copy and paste it, but you don't need to because it does it automatically. So you've got a little bit of an introduction to the data library. You've created a custom cohort of participants to study, and you've exported that or imported it, as the case may be, to your project workspace. Now, what can you do on it? So the next step is some analysis. There are two kinds of analysis you can do. Like I said, for the data that we just collected, we're going to do a notebook analysis. So you can click over to notebooks, and hopefully your runtime environment is ready to use, just like mine. I always go through first thing and I list the notebooks alphabetically because I don't love the default organization, which was most recently edited. But if you're actually doing a project in the workspace and you have a lot of notebooks, the one you most recently edited is likely the one you're actively working on. So that would be a really useful organization. So there are 4 notebooks in this workspace. The first one, I'm not gonna go through it all, but if you've never used Jupyter Notebooks, this is a great hands-on primer. It covers everything from how to edit a notebook to how do you run cells, and it has a few exercises that you can do. So it's a really good resource that I encourage you to run through really quickly if you've never used Jupyter Notebooks. If you have used Jupyter Notebooks, sorry, I had a note from my TAs who are waiting on the line if you do want to use them. If you have used Jupyter Notebooks, that's great. This will be a little easier for you to follow, but hopefully our notebooks have enough documentation that even if you haven't used notebooks before, you'll be able to keep up. So, as I said, when you run a new notebook, the first step is to create a runtime environment, which is a virtual machine or a group of machines cluster in the cloud. And just like when you bring a new laptop home from Best Buy, your runtime environment, when you first spin it up, does not necessarily have all the libraries and other things that you'd like installed in it. So oftentimes you have what's called a setup notebook. And these are no different. So if you click on, what I just did was click on the title of the notebook, and that opens it automatically. This is a preview, read only, but we actually want to run this notebook. So there are two modes. There's edit mode where you're running, you can run cells and save the notebook with the cells, with the code already run. Or there's playground mode, which allows you to run the notebook but not save it. And playground mode is useful if you're working with many people at once, because you don't want to have collaborators in different places overwriting each other's work. So only one person can be in edit mode at a time, and the person in edit mode is able to save changes, and everyone else working on the notebook is in playground mode. So we're going to select edit mode. And since my notebook runtime is active, it'll go very quickly. And this is what a notebook looks like. Yours may not have the table of contents on the left, and if that's true, you can click. There's a little menu item, there's a little button right below the help menu, the help tab right here, that will give you a table of contents. And it's useful in the way table of contents always is, in that it helps you know where you are in the overall notebook. If you have a longer notebook, it's really useful to be able to go back and forth. So just a useful piece. So let me introduce some of the big picture features of a Jupyter notebook. So the basic unit in a notebook are called cells. There are two basic kinds that we use. There's Markdown, which is documentation in this top one. And there are code cells. You see down here below the Markdown cells is a code cell. And you see this little toggle allows you to define which kind you're in. That's useful when you create when you insert a cell from scratch. You want to define whether it's a code cell or a markdown cell. So to run a cell, markdown, that just means to make it look nice, and for code, that means to actually execute the code. You can do it a couple ways. You can click the run button, which does exactly what it says. Or you can use a shortcut, which is Shift Return. And if you hit Shift Return on your keyboard, that has the same effect as running right here. Now, I said that this was running a virtual machine in the cloud, and it supports different languages, which are called kernels. This one is an R notebook, and you can see that at the top right here where it says R, or if you go under the kernel menu, you can change. The kernels that are supported by the Terra platform are a Python Spark kernel 2 or 3, Python 2 or 3, or R. And Jupyter notebooks are flexible in that you can write in the program language that you're most comfortable with. So you're not restricted to just one kind. What else should I say before we run? I'm going to run through. If you have questions, specific questions about how do I do this, or how do I, that you want to know, you should put them in the chat box, and Liz or Anton would be happy to answer your notebook questions, because I know if you haven't run them before, it can be a little harder getting up to speed. But once you run them, they're very straightforward. So this notebook is just a setup notebook, which means we just need to run it straight through. And you can do that by going up to the cell menu, and you can select Run All, and it will just run all the cells. You notice that cells that are not yet run or are in the process of being actively run will be in red highlight in the table of contents, and it will go through, and as they run, they'll turn white. So I'm going to point out, I'm going to go up, so Notice this first code cell has a 1 in it, and the second one has a little asterisk. So the 1 means that it has been run. The number is the commands that have been, the number of commands that have been executed in this session. So this was command, and it's all done. The second command has an asterisk, and that means that it's still running. When it runs, sometimes you'll get a pink output menu down below. Even though it looks like it might be scary, it's just some communication from the virtual machine that's running it, that's telling you some things. It's installing these packages in a different place because this variable is unspecified, and it's installing these dependencies. And it's taking a little bit longer than I expected, but that's how it works sometimes. Are there any questions while I'm waiting for it to run? I'm going to assume that Anton and Liz are doing a great job fielding those. So I'm a little surprised it's taking quite so long to run. I've actually run this in my workspace already, so I shouldn't need to run it a second time. So I might just quit out of this. Setup notebooks will, after they install these packages, the first time, when you restart your cluster, it just finished. When you restart your cluster, all those packages will still be there, so you only have to run it once unless you actually delete your cluster. So I noticed that mine has now finished. We've confirmed that they've loaded properly. And then we have some provenance. So if you go back and you want to know what version you used, what operating system you used, what language, all those things are reflected in the provenance. And that's useful both to colleagues who might want to reproduce your work, or to yourself if you happen to leave your notebook, your analysis, for a week or so because you're away or working on a different project, and you go back. The provenance will help you get back exactly where you were. Okay, so once you've run that, you can close out that notebook, and you'll go back to the Notebook tab, which lists all your notebooks, and we're going to run the notebook number 3, retrieve a cohort using BigQuery. And again, you want to mode. So this is a very, very simple notebook, and we've actually already done part 1. This is what I walked you through in terms of generating the cohort. So you can skip through down to part 2, which is some additional libraries that we are going to want for this particular project. You know, I've run this notebook before and I didn't clear it, so I'm going to go ahead and clear my output cells. So I'm just starting again from scratch. Install the library packages. So this next command sets up a program called FIS. You don't need to know the details, but what it allows you to do is to grab variables like the workspace name, which you name to be your own copy. This command allows you to grab whatever you've named it, and it will run it itself. And I feel like my cells did not clear. There we go. There we go. Okay. So since I cleared that successfully now, I'm going to rerun it really quick. So this is just some housekeeping setup. which you can read about, but you can just run them quickly. These steps are doing things like setting the project ID so that when we make our query, it knows how to bill you for the query that you're making. So again, this is a warning. It won't affect the output. So part 3 is where we get the cohort query that you made when you went to the Data Explorer. So it's a Python command, so we convert to Python, and then this actually reads the cohort query from the data tab of your workspace. This is we like to add sanity checks just to make sure you're getting the output that you expect. So this just gives a printout of the SQL query, and if we were to look back, this is exactly the syntax that Dave was showing. This next block is in case you have selected different inclusion criteria and you have different cohorts. It allows you to choose which one, but we're not going to do that. The next step is where we actually do the query. So we're going to call BigQuery and return the results in a memory table. And you can run that one, and you notice it ran very quickly. And let's examine the output. So the query just returns the participants of the table, right? So the participant IDs. And you notice there are 10 listed here. There's 240 more rows, and 10 plus 240 is the 250 participants that were in the study cohort that I created. So that's another sanity check. The next step is to do the join that Dave did in the Cloud Console. So this just is going to take those participant IDs that identify the people or the participants that you are wanting to use, and it's going to join with another table, and that's going to allow you to get either the metadata or the genomics data, with other data you want to add. In this case, it's just adding the gender to the participant ID. So we'll have a table of 250 participants and 2 columns. The first column is the participant ID. The second column is just the gender. And as an additional sanity check, we're just going to do a quick plot. So this was a really simple analysis, not particularly interesting, but in the Getting Started notebooks that are AMP-PD specific, you'll see where you basically are going to replace the last little bit with a more interesting AMP-PD analysis. So that is the notebooks portion. You can close that out. And the next hands-on part and last that I'm going to do with you is to run a workflow. So if you go to the workflows section of the workspace, you notice there are 2. So the exercises are actually outlined in the dashboard, but I'm going to go through the first one. So the workflows that are in this Quick Start workspace are file format conversion workflows, because everyone understands what a file format conversion is, and they run quickly enough that you should be able to get have a successful run in only a few minutes, which can be very satisfying. So the first one, if you click on the workflow, you can go in here, and you'll see you have a lot of parameters. These are the tasks of the workflow. This is a simple workflow. It only has one task. It's taking a cram to a BAM. The variable names are in this column, what type of variable, and the attribute is the actual value of the variable for this analysis. And there are a number that are defined. The disk size is 200. The memory size allotted is 15 gigabytes. Obviously, you can go in and edit these if you want. I'm going to leave all these the same. If we go down further, you notice there's a variable called input cram. Well, that is, as you can guess, the cram file that is going to be input for the workflow. And as an attribute, it has this funny prefix, which is this dot. And the prefix this dot tells the workflow, go to the data table in this workspace and select the variable cram. And that is where it will find the input data. And I will show you that again, but first we'll run through the actual workflow. There's also some reference files here, and these are also found in the data tab. And then the last piece in the inputs is the sample name. So this would be like the participant ID or whatever. And you see again, it has this dot. That tells you that, again, that's in the table that's associated in your workspace. So it's all integrated. You can look at the outputs from here. This particular workflow outputs 3 different things. It outputs an index file, a BAM file, and a validation report. Again, you notice that it says this dot in all of these, and what that tells you is that when the workflow generates output, it's the data file in your Google bucket, but it writes metadata to the data table. And we're going to run this workflow and you will see exactly what I mean. So we're going to select data. We're only going to process one row. So this is the data table that you saw when we went into the data tab, and the second row is actually a template. So if you wanted to run on your own data, you could go into the data tab and you could edit the sample ID and you could put your own cram file. But we're going to run on one that's already pre-run. So you click the row that corresponds to this sample ID NA12878, which is a public access cram file. And this is the location. If you can see, when I hover over it, it actually gives the full path for that data file. It's in a public data bucket. So we're going to select that data and then click OK, and then click Run Analysis. It asks you to confirm, because if you're running a big analysis, this could be a very expensive proposition, so it's going to ask you more than one time. It's going to tell you what compute engine is being used to process, and you can cancel if you wanted to do that, or you can click Launch. When it launches, you'll get the platform automatically takes you to the last tab, which is the Job History tab. And it gives information. Here's the data piece that you're analyzing. Here's when it was last changed, so a few minutes ago when I selected it. And the status is located as queued. So you've submitted your job to the cloud and it's waiting in line. If you want updates on this, you need to refresh the page. I'm hoping that it will run quickly, but it might not. How quickly things go from being queued to running depends on how many people are in line ahead of you, that sort of thing. So if you run, as I have, on a Saturday morning at 7 o'clock, they will go very quickly. that's all for my portion. I was hoping that it would run. And okay, so it was queued. Now it's been submitted. If you wait a few more minutes, it will turn from submitted into running. And if you wait a minute or two, it will convert into completed. And I'm not going to take up time redoing my window so that you can see that. But if you do it on your own, when it does finish, I'll do it one more time, but when it finishes, mine is running, so maybe I do have time. You can hop over to the Data tab, and if you click on the sample table that contains, so right now it hasn't finished outputting, but when the workflow is finished running, there will be additional columns of data in the sample table. So we started with this sample. Here's the sample ID. Here's the data, the input data that we started with. And when the workflow completes, there will be an additional column here that will represent the output data. I'm going to do one more quick look. It's done. So if you go over to the Data tab and click on the sample data, you notice the three outputs, the index file, the BAM file, and the validation report have been added to the sample table. So that is all for my part of the presentation. I'm ready to hand it over to Barry for the next part.